Hi, my name is Jason Gill, and I'm a member of the Center for Phage Technology at Texas A&M University. In this video, we're going to talk about the CPT's Galaxy and Apollo platform for whole phage genome annotation and analysis. The structural annotation workflow. The CPT's phage genome annotation process divides annotation into two major steps. First, structural annotation will predict the locations of protein-coding genes, TRNAs, and transcriptional terminators. The functional workflow analyzes the protein-coding genes identified during structural annotation to determine their function by a combination of similarity and conserved domain searches. In this talk, we're just going to be talking about the first part of this process, which is gene calling and the structural workflow. This is an overview of the CPT Galaxy structural annotation workflow. You can see here it's a pretty complicated process. This workflow executes a number of analyses starting with just your phage DNA sequence to start. By the end of this process, it will have an organism created in Apollo with the structural gene evidence applied to it as tracks, which will then be viewable by the user. I'm just going to go over some of the basic parts here, and we don't need to memorize the entire workflow here. Up here at the top, there are analyses that will identify tRNAs and transcriptional terminators. We currently use tRNAscan and Aridorn to identify tRNAs. We use two different gene calling programs. The first, shown here, is Glimmer3. This is a popular gene calling program and it's one used in NCBI, and this will identify protein coding genes from DNA sequence. As you can see here, the workflow will run Glimmer3 and then process its outputs to be viewable in Apollo. The other structural gene caller that we use is called Metagene Annotator, and again we are taking the tool output and formatting it for display in Apollo. The final structural gene calling part of the workflow here uses a tool called 6-Pack. This is what's called a naive reading frame caller. So this will just identify all possible reading frames that code proteins over a certain size. These are then filtered for only those that have a valid start codon, and they get formatted for display in Apollo. The last part of the workflow is the Galaxy Apollo bridge, which will transfer analyses from Galaxy into Apollo, where the user can view them in the context of their genome. This is an overview of what gene prediction tools Glimmer3 and Metagene Annotator will do. What these tools are doing is looking at the DNA sequence and predicting which reading frames actually encode proteins, and they'll display only those reading frames. What's shown here is the output from Metagene Annotator run against the phage lambda DNA sequence, and above that is a schematic of the phage lambda genome, which is very well characterized. You can see here that these gene prediction tools are generally quite accurate. This run of Metagene Annotator was able to correctly identify all the known protein coding genes, starting from gene nu1 through gene J, as shown here. It's worth noting that these prediction tools are only looking for normal protein coding genes. They can't find unusual features like, for example, programmed translational frameshifts or embedded genes as you might have with genes in span complexes. But as you can see here, in the case of lambda, it's done a great job and has accurately identified all the known protein coding genes in the first half of the lambda genome. So let's give a brief overview of what a prokaryotic gene looks like. A protein coding gene must have a valid start codon, and in our system, valid start codons are ATG, GTG, and GTG. These are by far the three most commonly used start codons in bacteria. The reading frame will encode a protein, and the open reading frame is determined by that start codon. The open reading frame is also commonly referred to as a coding segment, or CDS, which is, for example, how they're stored in GenBank files. At the end of the reading frame, there'll be a stop codon, which terminates the frame. Also upstream of every protein coding sequence, there should be a ribosomal binding site, which initiates translation. So that ribosomal binding site should consist of a Shine-Dalgarno sequence, followed by an appropriate spacer, followed by the valid start codon. The RBS, shown in blue here, has a Shine-Dalgarno sequence, shown here as AGGT, a short spacer, and then the start codon. All protein coding genes must be translated from an mRNA, which requires initiation at a ribosomal binding site. The RBS consists of a Shine-Dalgarno sequence, a 3 to 17 base pair spacer, and a start codon. The Shine-Dalgarno sequence will base pair with a complementary sequence at the 3' end of the 16S RNA to initiate translation of a protein. The strength of that translation is affected by how close the gene Shine-Dalgarno is to the consensus Shine-Dalgarno sequence, which is AGG-AGGT, as shown here in the DNA form. Any subset of that sequence could be used as a potential ribosome binding site with appropriate spacing. So on the right here, the box shows potential valid Shine-Dalgarno sequences. In the cell, translation is initiated for messenger RNA. But since we're looking at this in the context of genome sequences, it's shown here as how it appears in the DNA sequence, since all annotations are conducted on DNA sequence, and DNA sequence is what you're going to be looking at all the time during this process. On the left part of the column here, you can see all the valid Shine-Dalgarno sequences based on the normal Watson-Crick base pairing rules, the consensus Shine-Dalgarno sequence, and also valid subsets that could be used for translational starts. We have to keep in mind, though, that because translation actually occurs at the RNA level, wobble base pairing rules also apply as shown in the right column, where GU base pairs are a valid RNA base pair, allowing some variance in the Shine-Dalgarno sequence. At the CPT, we developed a tool called ShineFind, and we run this tool on all of our structural gene caller outputs. This tool looks upstream of each predicted reading frame, as called by MGA, Glimmer, or 6-pack, and identifies any potential Shine-Dalgarno sequences, and makes these a part of the gene feature as shown below here. The Shine-Dalgarno sequence will be appended as a small subfeature upstream of the predicted reading frame. When this gene is called, the Shine-Dalgarno sequence is part of the gene model, which is called and will appear in the user annotation track in Apollo. A note here on how phage genes tend to be organized. Phages tend to have very high coding densities, typically in the range of 90% or even higher. These features are very tightly packed, and they may actually even overlap by a few codons if that's possible. Phages, because they have to package all their DNA into a head of a fixed size, are somewhat constrained for space, and coding densities tend to be high in phage genomes, much higher than you would see in the average bacterial genome. Phage genes are also typically translated from polycistronic mRNAs, which means they tend to appear in large polycistronic blocks of genes. So we're looking at a gene arrangement here, and in this example, this is the phage lambda genome. You can see the blue and green blocks indicating genes in the plus and minus strand respectively. As you can see here, most genes tend to appear as collinear blocks either on the plus or the minus strand, with very little switching between the plus and minus strands for individual genes. This is an example of output from the structural workflow in Apollo. The outputs shown in these two tracks are from metagene annotator and Glimmer 3. These will detect most of the protein coding genes in your phage genome. The 6-pack evidence track will show all possible reading frames with a valid start and greater than 25 codons in length. All of these outputs have already been run through ShineFind to add any Shine-Dalgarno sequences associated with those reading frames. Most of your gene calling should be done from the metagene annotator and Glimmer tracks, but 6-pack can be used to identify genes that are missed by those gene callers, or if the gene callers identify a correct reading frame but an incorrect gene start. 6-pack can then be used to identify perhaps the same reading frame with a better gene start. ShineFind helps the user to identify good gene starts by identifying starts that are associated with Shine-Dalgarno sequences, which the majority of phage genes should be associated with. Sometimes, when reviewing your evidence, there will be disagreement between the gene calling tools. Here, Glimmer 3 has called a gene in this position, while metagene annotator has not. The call by Glimmer 3 fits neatly in the space between two adjacent genes, without excessive overlaps or gaps, and has a detectable Shine-Dalgarno sequence. In this case, we would call the gene from the Glimmer 3 track. Here, Glimmer 3 and metagene annotator have called genes in the same reading frame, but with different gene starts. The metagene annotator call has a good start, which is embedded in the upstream gene by about 25 codons. The Glimmer 3 call starts later, and has no detectable Shine-Dalgarno sequence. If we look at the 6-pack track down below, there is another start with a Shine-Dalgarno sequence that is embedded in the upstream gene by only 4 codons. In this case, we would prefer to call the gene from the 6-pack track. The program's metagene annotator and Glimmer 3 will detect most protein coding genes. Use the outputs of these tools first before resorting to the naive ORF caller 6-pack. ShineFind will automatically detect Shine-Dalgarno sequences upstream of predicted genes as a means of quality control. The majority of genes will have a valid Shine-Dalgarno sequence associated with them. To call genes from evidence tracks, Evidence tracks contain tool outputs, but these are not a part of your genome. Only features in the top yellow user-created annotations track are in your genome record. To transfer the predicted gene to your genome, right-click on it and select Create New Annotation, and then Gene. Another function of the CPT structural annotation workflow is to look for TRNAs. We currently use the tools Aragorn and tRNA Scan SE to look for possible tRNA secondary structures. These appear as separate tracks in Apollo when the workflow is complete. As with protein coding genes, it is advised to only call a single tRNA gene at a given position. There's a lot less to interrogate with tRNA calls. It is generally recommended that if it is called by either tool, it should just be called as a tRNA feature. Many phages have no tRNAs at all, especially phage genomes that are less than about 50 kb. So in many cases, the track will simply be empty for your phage genome. When calling tRNAs, make sure to add them to your annotations as tRNA features and not as genes. Click Create New Annotation and then tRNA to call tRNA features. Another part of the structural annotation workflow is the identification of row-independent terminators. Row-independent terminators form a stable RNA hairpin that will dislodge the mRNA transcript from the RNA polymerase active site. And these occur at the end of the transcript too, as the name implies, terminate transcription. To find these, we use a tool called TRNAs. So on the top left here, there's a schematic of what the stem loop looks like, followed by a poly-T tail. On the lower panel on the left, this is a schematic showing what's believed to happen to terminate transcription, where this hairpin snaps shut as it leaves the exit channel of the RNA polymerase, which then causes the transcript to be dislodged from the RNA polymerase active site, which is assisted by the poly-T tail, which has weak base pairing. So what we're looking for here is a short hairpin loop which is followed by a region of multiple T residues in the DNA sequence. These will also appear in the Terminators track in Apollo. So evaluating your Terminator output. The TransTerm tool provides a large number of false positives that need to be filtered out. It's bad practice to actually annotate every possible Terminator that's identified by TransTerm. The user really should be looking at this output window shown left for each Terminator, which you can bring up just by clicking on the Terminator in the evidence track. So we advise that Terminators only with scores above 90 are called. Users should look at the 5' and 3' stems. There should be at least 5 base pairs with no mismatches, and the downstream 3' tail should contain at least 4 Ts. The user should be very suspicious of low scoring or out of context Terminators. This is what we mean by out-of-context terminators. On the top panel, you can see these are all terminators shown as the black box with the arrows, terminating plausible transcripts. For example, they could be directionally in the same register as nearby genes, or they could be located at a position of convergent transcription. Basically, a terminator which is located near the end of what appears to be a transcript are more likely to be valid. In the bottom panel, these are examples of terminators that do not appear to be associated with any actual transcript, and so therefore it is not recommended that these be called. When calling terminators, make sure to add them to your annotations as Terminator features. Click Create New Annotation and then Terminator to call terminators. When actually annotating genes in our workflows, we suggest the following order of operations. We suggest that tRNAs be annotated first. This is relatively quick to do, and many phage genomes don't have any tRNA genes at all. It's important because tRNAs may actually occupy part of the DNA sequence that will then exclude protein coding genes from being called there later. The second step is calling protein coding genes, using metagene annotator and glimmer as the main gene callers, supplemented with naive ORF calls from 6-pack. And finally, terminators can be called. Once the structural protein genes have been identified, you have now the context for calling terminators. So the terminators that make sense can then be selected based on your gene calls. For more information on using the Galaxy platform, we encourage you to review the extensive Galaxy training resources that are available online. These include those available at the main Galaxy homepage, Galaxy 101, and of course the Galaxy training materials produced by the Center for Phage Technology, which are specific to our system and located at cpt.tamu.edu/training materials. The CPT training materials contain detailed instructions on how to upload and manage your genome in Galaxy, and how to run the major analysis workflows. If you use our system, we also encourage you to cite our publication in PLOS Computational Biology that describes the platform and the associated workflows. The CPT Galaxy Apollo platform is supported by the National Science Foundation, Texas A&M AgriLife Research, and Texas A&M University. We would like to thank the many programmers and scientists who have helped design and build the system, and the excellent and supportive Galaxy and Apollo user communities.